Hi, General.
Hi,
Hello.
hi.
Hi, General.
Yeah,
Yeah.
I can hear you.
Can you start to invite Professor Bingham to give opening remarks?
Okay, okay.
Hello,小峰。
Hi, 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 Bing.
Hi，小峰。
Hello,
Okay.
I was.
Ron
Hello,
will
everyone. Welcome
the
to. Okay.
okay. Please start.
Yes, I will make a brief introduction and invite Dr. Bingham to give the opening remark. Okay. Hello, everyone. Welcome to the RSPS 2024. That is Rising Stars in Plant Sciences 2024. My name is Rong Chen, a scientific editor from Molecular Plant and Plant Communications. It's my great honor to host the first day session of RSPS 2024. First and foremost, please join me to welcome Dr. Bing Han, the co-editor-in-chief of Molecular Plant and editor-in-chief of Plant Communications, to give an opening remark. Please.
Good evening, everyone. I'm delighted to welcome you all to the oral presentation contest featuring the finalists of Rising Stars in Plant Sciences 2024. The RSPS Award was established in 2022 to honor exceptional early career plant scientists and inspire them, along with the future generations, to engage in cutting-edge plant research. Over the last 2 years, we have recognized a total of 9 young scientists as winners. This year, we announced and opened applications for RSPS 2024 in early January. Currently, 8 outstanding young scientists have been selected as a finalist by the scientific committee. Today and tomorrow, they will deliver online presentations showcasing their scientific accomplishment and new research discoveries. Subsequently, the committee will conduct final evaluations and vote for the winners of RSPS 2024. I take this opportunity to express my gratitude to the Molecular Plant editorial team and the scientific committee members of RSPS 2024 for their dedicated time and effort in making this finalist oral presentation contest possible. I also extend my thanks to all the finalists for applying to RSPS 2024 and agreeing to share their exciting research through oral presentations today and tomorrow. I wish each of you success in your presentations. On behalf of the Center for Excellence in Molecular Plant Sciences at the Chinese Academy of Sciences, as well as the journals Molecular Plant and Plant Communications, I welcome you all to participate in our upcoming conferences and events. The global plant science community continues to expand, especially in China, where many young PIs and students are eager to exchange scientific achievements and ideas with colleagues worldwide. We are committed to organizing international conferences and the RSPS Award in the years ahead to advance plant sciences. So together, we can accelerate the progress of global plant sciences and create a bright future. I hope you enjoy the presentations by the RSPS 2024 finalists. Thank you all. Thank you very much.
Thank you, Bing. Thank you for your introduction to the SPS and the warm opening remark. Now let's take our scientific journey to SPS 2024. Today we will have 4 excellent finalists. Before we start, a gentle reminder that each finalist will have 22 minutes for oral presentation and 8 minutes for Q&A session with the scientific community members. So now let's get started. The first speaker is Michał Bandrowicz from Poland, and he will talk about production of trees in overheated world. Now please join me to welcome Michał.
Hello, good morning to
Hello.
everyone. So first I will try to share my screen. This worked 1 hour ago, should work now. Okay, I hope that you see
Yes,
my
we can.
screen. So first, thank you very much for having me here. It's a great honor to be selected as a finalist for this amazing competition. And well, without wasting any more time, I will start to talk about what we are doing here, and I will talk about climate change and the reproduction of plants, trees especially. This is the topic that we are doing here in Forest Biology Center in Poland. You've probably seen many of those pictures over last years. Forests worldwide are under increasing pressure from climate change, which results in these large-scale disturbances that are killing trees on large scales, which we call forest diebacks. This is just one picture from my colleague in the United States showing burned forests, but you've seen those also from Canada and Australia over the last few years when we had these mega fires killing forests. And in general, the forest diebacks and most likely due to climate change are increasing all over the globe. This is a study from Europe showing how it is accelerating over the last 4 or 5 decades. The graph on the right shows the essence of all of it. The area that is prone to disturbance has tripled in the last few decades and is projected to increase further. The key message from what I'm saying here is that the forests are dying at the increasing rate. The mortality rate is increasing, and therefore we need seeds for the new generation of forests, because forests are important for biodiversity, for carbon sequestration. They provide major economical and societal services to humans. And at the same time, even though we need those seeds, we know very little about generally about this, the reproduction ecology of trees and about the climate change effects on this in particular. And just to show you how large is that gap in knowledge, I did a very quick Scopus search in which I've put those following keywords. So either climate change, trees, and then growth. And this produces quite a lot of papers. So we know how climate change is impacting growth of trees, climate change, trees, and survival. So again, we know quite well how mortality and survival of trees is being impacted by climate change. But then if I put recruitment on seed production, we have very small number of papers, which reflects a more general pattern of tree ecology, in which we know a lot about how trees are growing, how trees are dying, and very little about how trees are being born. And one more illustration of this, how little we know about seed production ecology. This is a picture of a Quercus ilex. It's an oak growing in Mediterranean areas. This one is from Spain. I did that picture a few years ago when we were doing fieldwork there. And, if we would ask a scientist or a forester about how fast this tree is growing, not this individual in particular, but in general, Quercus ilex, then we can either know or find the answer. We know the annual rates of growth. We know how climate is affecting that growth. So when it's dry, how the growth would be declining, and so on and so on. But if we would ask exactly the same question about fecundity, so the number of seeds that are produced by that species, there is no one that could answer that question. So a very basic question, like how many seeds on average this species is producing? Is it more fecund than some other oaks that are growing nearby? We have no idea. The basic questions remain unanswered. And one reason for why this is so difficult to answer those very basic questions is mass seeding. Mass seeding is a reproductive behavior of perennial plants, trees in particular, in which plants are producing seeds not in a regular manner, so we do not have more or less the same number of seeds each year. Instead, seeds are produced on intervals. So we have years when there are no seeds, and years when there are thousands of seeds produced by all individuals in population. So this is synchronized. We have either no seeds at all or many of them, to simplify. And this happens all across tree of life. So for example, on the picture here, we have alpine grasses from New Zealand, and you can see this boom and boost dynamics of seed production. We have Pinus sylvestris from Europe, or Fagus sylvatica trees, and so on. So, and because of this large interannual variation in seed production, it's quite difficult to actually know the average seed production, because it's not enough to just go 1 year in the field and count seeds, because you may happen to count in a low year or very high year, also middle year. So you just need to have a long-term data set that will count seeds for many years across all individuals to get an idea of what those plants are actually doing in terms of seed production. And as I said, this reproductive strategy is prevalent across plant kingdom. So this is a picture from a study we published last year, and you have like around 600 species here, and in red are species that have high interannual variation in seed production. So those that are that must strongly. And as you can see, that the red color is present all across the board. And there are, of course, families like Finalist or Fagales in which masting is more frequent. But the point is, masting is a prevalent reproductive strategy across plant kingdom. And, well, it makes it a little bit more difficult to understand ecology of reproduction of trees and perennial plants in general. And before I will go to the next topic, I will just also show you something that is very impressive in that reproductive behavior. This is the geographical scale of synchrony. As I mentioned before, the interannual variation in seed production is synchronized across individuals within populations, but also very often across populations. So here on that graph, you have Europe, and this is data on seed production of European beech. It's a major forest-forming species in Europe that has a very large economical and social importance. This is a graph done by my friend Kuba Szymkowiak, and big points are showing showing years in which there were lots of seeds, while small points are years when there were no seeds at all. So as you can see, that variation, so years when there are no seeds or years where there are lots of seeds, are synchronized across basically the whole continent. So imagine how important it is for food webs functioning and so on. So there are either years when forests are silent, there is no food at all because the trees are not producing seeds, and all those seed consumers that rely on seeds have just nothing to eat. Or you have years in which millions of trees across huge areas are producing millions of seeds, which then produces boom in population dynamics of seed consumers. So the question is, if masting is so frequent across the tree of life, the question is why plants do it. And there are two major reasons for it, and this is either predator satiation and pollination efficiency. And let me explain what does it mean. So predator satiation is all about reducing seed predation. Plants, trees are producing very often large seeds that are nutritious, so there are lots of animals in the forest that will be keen to consume them. At the same time, plants cannot just run off from seed consumers, so they need another strategy to protect their seeds. And actually, a very effective one is just to starve out all the seed consumers from the area. So if there is a year when there are no seeds being produced and you are especially seed consumers, well, you die because you starve. And if that famine created by masting continues, it effectively kills all the seed consumers in the population. And then when the mast year, a huge seed production happens, there is no one around in the forest to eat those seeds. And this is a very effective anti-predator strategy. And the other thing that that masting is doing for plants is increased pollination efficiency. Many masting plants are wind pollinated, and because masting allows to concentrate flowering in one huge event, which basically saturates air with pollen and increase pollination efficiency. Wind pollinated plants do not have animals that would carry the pollen to the flower, so therefore they just saturate the air with pollen using masting for this. So this is why masting happens. And the second question is how it happens, and so what's the physiological mechanism behind? And in like, to shortly speaking, one major mechanism is sensitivity to weather variation, which we call in our field weather cues. So this is a graph showing seed production again in European beech, the species that I already mentioned. And this is the response of European beech to variation in summer temperatures. Basically, how this works in European beech is that if there is a cold summer that is followed by hot summer, so that's why we have delta T here, so the difference between temperatures in one summer and another, then the tree is producing lots of seeds. Whereas if the cue has not happened, they are not producing them. So by using the weather cues and being sensitive to those weather cues, they can produce those large interannual variation in seed production that is synchronized across individuals because weather is synchronized across large scales. And that's important for climate change, to which I now will go, because if you are dependent on weather variation and you use hot summers to trigger your reproductive event, then, well, it's likely that you will be impacted by climate change. And this is what we are seeing. And we already know that warming breaks down mass seeding. And I will explain now what does it mean. So again, going to European beech, the study species that we are studying here a lot. This is a data set in which that monitors seed production of European beech in England, in the United Kingdom, for now over 40 years. And what you can see here, each gray line is one individual tree, and the blue line shows the average across all the trees that have been monitored. This is about 15 populations and 150 trees across England. And as you can see, in the 80s, this was as expected. We have these years when there are lots of seeds, and then years when basically none or actually no seeds are being produced at all, like for example here or here. And then something happened in around 2006, and this all became a mess. Instead of having this very nicely synchronized seed production patterns across individuals, we basically have a spaghetti plot here. So the synchrony collapsed. The trees are no longer well synchronized. And also you can clearly see that there are no more years when there are no seeds. There's always some trees in the population that are producing seeds. So masting is no longer variable and no longer synchronized. And if you think for a second about the predator citation mechanism that I mentioned a few minutes before, that's important. And I will go back to this in a second. So the first question you can ask is, well, how it happened that the pattern of seed production has changed so much? And the reason is warming. So in our population in England, in the last 40 years, the summer temperatures that are important for triggering masting on average are warmer by 1 Celsius degree. And this 1 Celsius degree warming translated into way more frequent triggering of flowering years. So this is the red line here, and this is the probability of a hot summer that would trigger masting. So in the past, it was about 10%, and now it's about 50%. So trees are being forced by weather to reproduce way more often now than they were in the past. And therefore, they don't have the resources to respond to this. Like, you know, in the past, there was a trigger, they reproduced, and then they were resting and accumulating resources for another big seeding event for 2 years. Now, basically, they are being triggered all the time, and they just cannot keep up because they do not have the resources to do so. And this graph nicely shows what I am talking about. So here on the left, we have the relationship between seed production and weather cue in the past, before the breakdown. And as you can see, seed production happens when cold summer is followed by hot one. Every time when the cue happens, trees are reproducing, then there is some time of a break, and when it happens again, they can produce seeds again. But what happens now is that because and importantly, you don't see years when there will be a cue and like a hot summer and no seed production. Whereas now, because those hot summers are so frequent, and you can look at the bottom right corner of the graph on the right, even though the queue happened, trees are not producing that much seeds or not at all, just because they do not have resources to do so. And this breaks down both synchrony and the internodal variation in seed production. And you can say, well, that's just the more regular reproduction. What's the problem? Well, the problem is that the internodal variation and synchrony is this crucial strategy to reduce seed predation and increase pollination efficiency. And weak masting is no longer an effective reproductive strategy. And so for that population of European beech that we have been following for the last 40 years, we also have data on seed predation rate by insects and on pollination efficiency. And let's start from the graph on the right. So this is average predation rates across all the populations that we have been studied. And you can see that in the 80s, when masting was doing what it should be doing, so variability was high and synchrony was high, masting was amazingly effective reproductive strategy. The predation rate were about 1 or 2%. So they were not losing almost no seeds to seed predators, seed consumers at all. Whereas now, because there are always some trees in the population that will be producing seeds, the starvation is not working. So the population size of the insect has increased a lot, and therefore every second seed is being eaten of those that are being produced. So let me repeat, from 1% of destroyed seeds to every second seed that is being now destroyed by insect, because the variation and starvation no longer operates as it was operating in the past. And similar, although a little bit less spectacular, stuff happened to pollination efficiency that decreased from about 50% to less than 40%. So the question is how that matters for reproductive potential of those trees. And well, now we are looking at the seed production which is a long-term average to get rid of those high peaks and downs. And in green line, we have seed production of a large tree, and in the red line is the seed production of a small tree. And if you look just at all seeds, so including those predated and so on, nothing basically happens. Those trees are doing fine as they were doing 40 years ago. Maybe there is a slight increase which is associated with the fact that the trees has been growing, so they are a little bit larger now than 40 years ago. But if we take out seeds that were consumed by insects and seeds that were unpollinated, then what we see is actually a collapse of reproductive potential of European beech in England. So now we have big tree in green and the smaller one in red, and you can see that in the big trees, the production, the fecundity, so production of seeds that can actually lead to another generation of trees decreased 5-fold, which I think is a collapse of reproductive potential. So the question is now how this will impact regeneration and whether this way smaller seed supply can actually sustain generation replacement. But well, one can say that we have been studying one island, England. So the question now, the larger one, is whether this phenomenon of climate change, warming impacts on reproductive patterns in European beech is something that is happening more widely, or maybe this is just restricted to those populations that we happen to study. And just a few months ago, a study that we've been doing that has been led by Jessie Forrest, a PhD student from University of Liverpool. We have collected data on seed production of European beech from basically majority of the species range. Those dots are showing populations of European beech on which we have the data. And the background, the red color, is the rate at which summer temperatures are getting warmer. And that's already important. Basically, across all the species range, summers are getting warmer. And as I explained before, hotter summers are a bad sign for masting in that species. So then we looked at whether the frequency of the cues, of the triggers of masting increased across the range. And yes, we found that because warming is accelerating, and the same happens with the triggers. So this is again the summer temperatures through time, and we can see that the positive anomalies, so the probability of occurrence of hot summers, has increased from less than 10% to about 30%. So it means that trees in European beech trees across whole Europe are now triggered more and more often for seed production. And then the final bit is whether this decreases CV, so the internal variation of seed production, so whether breakdown of masting of European beech is occurring across the species range. And yes, it is. So now we have the CV, which we use as an index of internal variation of seed production, and this is June-July temperatures. So if populations are experiencing increased summer temperatures, the masting is being less intense, and also if the rate of the change of the temperature is higher, then the rate of masting collapse is higher as well, which signals that reproduction of that species is a danger basically across the whole species range, especially in places where summer temperatures are increasing at rapid pace. And then a few final bits. One final bit is one graph from a paper we published 2 years ago on predator satiation. In this study, we have collected data from around 60 studies and a few dozen more species. And one of the questions we asked there is whether predator satiation, so that mechanism that is decreasing predation rate, is being less effective over time. And it is. So on this graph here, we have seed production and here time, and in colors, we see predation rate. And as you can see in 80s, masting was very effective. So predation rate were decreasing from 50% to less than 10% as seed production was increasing, but it is no longer happening in the current time. I don't know whether in this particular study this is climate change, but the fact is that predator satiation is less and less effective, and therefore seed supply is is smaller. So to summarize very quickly what I've been saying for the last 20 minutes, seed production ecology lags behind mortality and survival, which is a bummer because we need seeds to replace the trees that are dying. Masting is widespread, and we need to understand masting ecology to really understand how climate change is impacting the seed production and regeneration potential of trees. Those complex interactions that follow from masting breakdown lead to declines in fecundity that we would not see if we just focus on seed count. So we need to understand those processes behind masting to really understand the impact of climate change on fecundity. And the questions are, how widespread is the masting breakdown? I've just been showing mostly European beech data, but there are studies that are being published right now that are showing that the same mechanism, the same phenomenon is happening in US, in Japan, in New Zealand. So it suggests that seed supply problems may be actually a widespread problem across ecosystems. And then we'll need to understand the consequences of that. What are the consequences of short seed supply on recruitment, on food webs, and whether we can develop forest management strategies that can help us to, buffer those changes. And that's all. Thank you very much.
Thank you, Michał. Thank you for interesting talk. Here comes the Q&A session. So committee members, if you have any questions for Michał, please feel free to open your microphone.
Thank you.
Any questions? Yes, Isabel?
Yeah, hi, very nice presentation. I have a question from a developmental biologist background. So at what point is this decided, this masting? It has to be something with already the initiation of the flowers, and how far is this correlated with vegetative growth? So with how many new leaves and branches there are.
Okay, so the process is being decided across many years. So as I mentioned, for example, in European beech, it appears that the plant is reading the environment and deciding across that time for 2 years. So this is cold summer, hot summer, then flower initiation, which is probably some molecular mechanics that we don't really understand yet, and then this leads to more flowers being produced. And then the second part of the question is about the vegetative growth. So, for example, in European beech, this is very interesting because if they are producing more seeds and flowers, it means that they produce less leaves, because that's like a competition for space on a tree crown. And therefore, they in very big years, when there's like these branches of trees are really like overwhelmed with seeds, they are just full of them, they will have less leaves. The crowns are sparse, and the photosynthetic rates are smaller, and the growth of trees is reduced by about 50% in those years. So you can see that, for example, if you look at the tree ring records of like the annual variation in tree ring records, then in years of seed production, there is a large depression in growth that is of comparable size to years of the depression when there is a strong water deficit.
Thank you.
Thank you for your question.
Thomas?
Yeah, hello. I was wondering, Samaru is also quite far from tree ecology, seed production ecology. I was wondering how you measure seed viability, and also how you obtain your data. So seed viability, of course, we would test germination efficiency, something like that. Do you measure the numbers of seedlings that you find, or how do you do it?
So germination trials would be great, but having them over 40 years would be difficult. How this particular data set is being collected is quite simple, because you need simple methods that can be maintained over 40 years across large scale. So basically, we go to the forest, we have either seed traps, so like buckets under trees to which seeds are falling, or we collect seeds from the ground during the constrained time frame. So I think here is 3.5 minutes, and you just collect them and bring them to laboratory, and then in the laboratory we basically sort them out, and that's very easy in European beech, because if there is a larvae inside, you will either find a larvae, or there will be a hole through which the larvae has emerged from the seed, so then it's empty inside. And European beech is a very nice model species, because when flowers are not pollinated, they still produce seeds, but they're just empty inside. There is no embryo. They just produce the husk. We don't really know why they are doing this. Maybe this is something to help with the rodent predation. That's something that needs to be studied, but we will, thanks to this, from seed counts, we actually know the pollination rate.
Yeah, very interesting.
Thank you.
Thank you.
Thank you. Any more questions? If no more questions, thank you again for your talk, Michal. Let's move on to the next speaker.
Bye-bye.
Dr. Hao Dongchen. Bye-bye. Dr. Hao Dongchen from Tsinghua University, China. He will talk about how do plants sense and respond to gravity. Please join me to welcome Hao Dong.
Hi, so can you see the full screen?
Yes, not full screen. Yes, now, okay.
Okay. Okay, so hi everyone. It's a great pleasure to give a talk here, talk about how do plants sense and respond to gravity. Actually, the gravity acts on all the organisms on the Earth, but the plants have a specific response to the gravity. You can see when the seeds germinate in the soil, they growing from all kinds of directions, but within a very short time, the roots will adjust to growing down. We call this positive gravitropism, and the shoots they will adjust to growing up. We call this negative gravitropism. So the gravitropism is essential for the survival of the plants. only after this adjustment, the shoot can penetrate the soil and growing out, then start photosynthesis. So on the Earth, the gravity's intensity and direction are almost constant. When we talk about gravity sensing or gravity stimulation, we talk about the direction. Means that when plants sense it not in the gravity vector, they will change their growth directions. Here is the one of the photos I took in the seaside in Hainan province in China. The strong wind blew down the tree, but the shoot can grow up again. So in the lab, we just reorient the plants to mimic and to trigger the gravity stimulation, and they observe the growth response of root and shoots. So the gravity controls the growth directions of different organs of the plants, and then further affect the architecture and the agricultural trace. Here is one of the examples to show that these two rice varieties, there's only one gene difference between them, to show that this right one show better gravity tropism. Thus, in drought condition, this rice can absorb the water in the deep soil, that's growing much better than the control and then yield better. So actually in the field of gravity sensing or gravity tropism, there's a long history. In the 1880, Francis Darwin reported these studies in his famous book, The Power of Movement in Plants. He found that the tip alone can act and respond to gravity, and then transmit some influence to adjoining parts to cause the bending. The influence was later to be demonstrated to be auxin. Then in the early 20s, Francis Darwin, the son of Charles Darwin, together with other two German scientists, Niemek and Haberland, they proposed the starch statolis hypothesis. They found that in the endodermic cells of the shoot and the columella cells of the root, they all contain aminoplast. Aminoplast means those plastids that contain starch. Their density is higher than other components in the cell. That's they always moved down to the bottom of the cell. So they think this sedimentation of aminoplast can initiate gravity sensing. So during the last 120 years study, the gravity signaling have this kind of frame, the gravity sensing, signal transduction, and the growth response. The starch statolis was proposed to explain gravity sensing. And Claude and Wendt, this is the last name of two scientists. This theory was proposed to explain the signal transduction. The auxin always accumulate in the lower side of the root and shoot. Then thus the asymmetric distribution of auxin will trigger the bending of the roots to growing down and shoot to growing up. So in this field, there's a long-lasting question. Although we observed the aminoplast sedimentation, but we still don't know the physical information, how this kind of move down physical information converted into physiological signal. So in the history of the gravity study, there's also another group of genes called LASY, was first identified in maize in 1981. You can see that here the maize cannot grow vertically. They called show a prostrate habit. They called this name LASY. And after several years, also identify similar phenotype mutant in rice. But after many years, until 2007, this gene was cloned in rice first, and then demonstrated to play conserved roles also in maize and in Arabidopsis. This LASY1 controls the negative gravity tropism of shoot. And then in the recent years, this LASY family members 2, 3, 4 was found when they mutated together. They show gravitropic defects, and the different groups all get similar. The result of defects, but there are some small difference between them. And in our group, we knock out the 2, 3, 4 LASYs in Columbia background. We found that when these three genes was knocked out, the roots growing toward all different kind of directions, and we get different alleles show similar phenotype. If you put either one of the lazy genes into the mutant background, the roots were growing down. Means that these 3 genes work redundantly in this process. Then how does this lazy regulate auxin? So in the wild type plants, after reorientation, the auxin accumulate on the lower side much more than the upside. But in the mutant background, this asymmetric distribution has disappeared. Thus, we concluded that the lazy control gravitropism will mediate the polar auxin distribution. Then further, we check where are the lazy proteins localized. Excitingly, we found all these 3 lazies localized in the columella cells. Those cells was proposed to be the gravity sensing cells. And also lazy4 have some additional signaling in endodermis. And within the cells, we found very interesting localizations. The first is in these columella cells, we found that the lazy localized on the surface of aminoplast. Because aminoplast was proposed to be the gravity involved in gravity sensing, means that lazy may also be involved in this process. Also, we found on the plasma membrane, the lazy show a polarity. You can see in the cell, they always localize on the lower side of the cell. And for all these 3 lazy show similar pattern. So this is exciting because the polarity of lazy may indicate the direction of the gravity. We further use immunogold labeling to prove that lazy actually localize on the plasma membrane by binding the phospholipid. So now lazy have 2 localizations. One is aminoplast, one is plasma membrane. Are there relationships between them? We change the growth direction of the Arabidopsis root. And at the beginning, you can see that the lazy localize on the original lower side here on the left. After 20 minutes, the aminoplast moved to the new lower side, and the lazy is accumulated there. Very nice correlation. Here is the upside down movement, similar results. At the beginning, aminoplast here, and after close to 80 minutes, the aminoplast sediment to the bottom, and then the lazy proteins move from the upside plasma membrane to the lower side. So it means that the lazy always accumulate on the plasma membrane adjacent to the aminoplast. Then we further study whether the aminoplast sedimentation control the localization of lazy on the plasma membrane. We use a PGM1 mutant. In this mutant, there's no starch. That's the plastid, the density is lower and the sedimentation very lower. You can only look at this upside down experiment. You can see after close to around 2 hours, the aminoplast without the starch just moved down a little bit. Thus, the repolarization of lazy on the plasma membrane was much disrupted. So it means that the sedimentation of aminoplast is important for the redistribution of lazy proteins. Then we check whether lazy proteins can move between these 2 localizations. We bleach the signal of lazy on the plasma membrane of cell 1 and bleach the both aminoplast and membrane signal in the cell 2. And then we check the signal recovery. After 100 seconds, you can see that the signals recover very well in the cell 1, but not cell 2. So it means that the fluorescence protein can move from the aminoplast to the plasma membrane. So with this, we have a very nice cell biology data to tell that the aminoplast sedimentation can trigger and guide the lazy proteins to the new bottom of the cell. Then we ask the question, so how does gravity stimulation mediate the translocation of lazy proteins? Are there any biochemical activity changes of lazy protein during this process? Thus, we did phosphoproteomics analysis. This is the vertical plant, so we do the rotation. And in this process, the beginning and the end to growing down of the root and the shoot up again. So in this process, we found that at the beginning and at the end, the lazy protein was in the low phosphorylation states, and in the middle stages, the phosphorylation is higher. So it means that the gravity stimulation can trigger the phosphorylation of lazy proteins. Then we search for the kinase contribute to this process. Interestingly, at the beginning, we found there's two MAP kinase proteins. At the beginning, there's no interaction. After gravity stimulation, very strong interactions. And then after the plants growing up down again, there's no interaction. Very nice dynamic interactions. We in vitro experiment can confirm this interaction and all the phosphorylation mass spectrum size consistent with the in vivo data. Then we ask whether this phosphorylation has physiological significance. We mutate this phosphorylation size within lazy4, and then we check the localization of lazy4. We found that the original lazy4 localized on the aminoplast well, but after the mutation, within the cell was almost empty. There's no signal there, means that lazy cannot localize on the aminoplast again. Also, the polarity of the lazy proteins was dramatically reduced. And these mutated lazy proteins cannot complement the triple mutant very well. We also do the reduction of the MAP kinase proteins that result in similar effects. Okay, so it means that the phosphorylation of the lazy may enhance its interaction and localize on the aminoplast. How that can be achieved? Then we search for other proteins localized on the aminoplast. So in the literature, we found in Patrick Masson's group, they identifying this TOC protein. Actually, there are translocans on the outer envelope membrane of chloroplast, also on the surface of the aminoplast. The single mutant phenotype is very weak, but when combined with ARG1 mutant, they show very strong gravitropic defects. But the underlying mechanism was not clear. We after we found the lazy localized on the surface of aminoplast, then we thought probably these two proteins interact with each other on the surface. Then we checked the interactions between lazy and TOC proteins. And unfortunately, in the period, we could not detect any interactions until we identified the lazy's phosphorylation. And we use the phosmimic lazies to test the interaction. We found they interact with TOC34, TOC120, TOC132. These TOC proteins very well, also with their domains. These TOCs form one of the TOC complex. And the lazy didn't interact with any other TOCs and didn't interact with TOCs within the membrane. Only with this TOC complex, the surface toward the cytoplasm show very nice specificity. And also within the root, the lazy proteins can colocalize with the TOC proteins very well. Then we think if the TOC proteins are important for lazy localization, we knock out the lazy proteins, we will see some localization change of lazies. And that's true. When we knock out the TOC proteins, we can see that the lazy proteins cannot localize on the surface of aminoplast anymore. And also, after gravity stimulation in the control, they show the polarity on the lower side, the lazy proteins. In this mutant background, there's no asymmetric distributions anymore. Then we check the phenotype. We found that the white type after gravity stimulation for 12 hours, the roots growing down. But in the mutant background, the roots just grow toward the original direction, cannot show the nice gravitropic response. So put this information together, we get this kind of a model. When the plants was reorientated, the aminoplasts, because their density is higher, they were sediment to the bottom side of the cell. At the same time, the lazy was phosphorylated and enhanced its interaction with the TOC proteins on the surface of the plasma membrane. Then, during the sedimentation of the aminoplast, will bring the lazy proteins to the new lower side of the columella cells. Then, accumulated the lazy protein here will trigger the asymmetric distribution of auxin and further the bending of the roots. Thus, with this conclusion, we give a molecular interpretation for the starch statolysis hypothesis proposed more than 100 years ago. So in the same period, the Mio Morita's lab published a paper in Science with similar results. They show the lazy can translocate from statolysis to the plasma membrane. Thus, our results can support each other to make the conclusion to be very solid. Our group provided the additional biochemical data about the lazy phosphorylation and the interaction with TOC proteins. Okay, so after the publication, we got many comments to say that we made a great breakthrough in this field. And a very interesting picture used by the editor of the Nature Plants. They show that germinated maize seeds, although the roots germinated in the upside, they were growing down, and the calyptile germinated on the bottom were growing up. This is means that after the gravity sensing, the different organs choose different growth directions. That's also another question in gravitropism. It's about up or down. So for example, show here, when the plants was not in the vertical directions, the oxygen accumulation here in the shoot accumulate on the lower side compared to the upside. In the root also accumulate on the lower side, much more upside. So it means that the oxygen's distribution in the hypocortical and the roots are the same, but the gravitropical response are opposite. The shoots growing up and the roots growing down. So this means that the downstream signals of oxygen in these two organs are different. So once we use the soybean, because the hypocortical is thick, we cut the hypocortical to be upside and the lower side and do the transcriptome analysis. We identified a group of sour genes. These sour genes accumulate on the lower side much more than the upside. And after studies, we obtained this kind of model. During the vertical growth, these sour genes distribute asymmetrically on the two sides. But after they are not in the vertical direction, the oxygen will show asymmetric distribution, accumulate much more on the lower side. Then the oxygen will degrade IAA proteins, release the alpha transcriptional factor. These transcriptional factors activate the Sol-19 genes expression. This group of the genes have the function to promote the cell elongation. Thus, the lower side will growing faster than the upside, the trigger upside growth of the shoot. And in our lab, we use the Arabidopsis to study the gravitropism. At the same time, we use the Phascomatrella because it represents a different type of gravitropism. All the gravity sensing and the gravitropical response happened in the same cell, the tip cell. Also, the Phascomatrella belong to the moss, belong to Bryophyte, the earliest land plant. That during the landing, the gravitropism build up and become better and better. That's with these studies may provided some evolution in size for gravitropism. So also we use the stage of the gametophyte of the moss. They are proteinema growing up, and this stage is kind of haploid stage. That's the mutagenesis can generate mutants very easily. So after mutagenesis, we get many mutants growing down, just to show the original upside and here growing down, change the growth directions. We get many of this kind of mutants. And here I introduce one of them. We cloned the gene and we found it is a kinesin motor. And when we labeled the motor and the microtube, we found that this motor can move along the microtube towards the minus end and far away from the tip and transport some cargoes to control the gravitropism. Due to the time limitation, I will not go into the details, but I'd like to emphasize the significance. The microtube has long been considered to be involved in gravitropism. People use the chemical to treatment, disrupt it. The gravitropism was show the defect, but there's no factors specifically involved in this process. So here I provide direct evidence for the involvement by identifying a specific kinesin motor. Okay, so here is my summary about the gravity sensing in plants. We found that the aminoplast sedimentation may repolarize lattice to achieve gravity sensing. That's provide an interpretation for the statulus hypothesis. For the gravitropical response, we found that the asymmetric expression of soil genes may promote the gravitropism in hypocotyl, and also we identify a kinesin motor and direct the gravitropism in Phascometrella. So in the future, I will continue to study the gravitropism, mainly focus on these 4 aspects. The first is the novel mechanism of gravity sensing. We have some evidence besides the aminoplast sedimentation, there are other sensing mechanisms. And the second is the growth direction of plant organs. The different organs growing different directions, how were they controlled? And then the evolution and agriculture application on the Earth, and probably in the future other planet. Okay, I'd like to give my thanks to my group members previously and currently, and also specifically give my thanks to Professor Xinwan Deng, my supervisor for PhD and then postdoc training, his lab, and worked there several years. His research interest is light signal, but allowed me to do some gravity signal studies and bring to Tsinghua University. I appreciate his help a lot. And thanks to the collaboration of Dong Juan in the LASI project, and thanks to David Cove and Andrew Kami, University of Leeds, they are pioneers in use the Phascometrella to do the molecular biology studies and give me a lot of support about material and techniques. Okay, thanks for your attention. I'm happy to take questions.
Thank you, Hao Dong. Thank you for interesting talk on gravity. It's time for Q&A. Committee members. Yes, Li Guoshan.
Yeah, hi, Hao Dong. Yeah, it's really fascinating talk. Congratulations for the nice work. I have a couple of questions. First, in the beginning, you saw the lazy mutant, multi-family member mutant, so the defects for the gravitropism, right? However, when you express one member, either 2, 3, 4, I cannot remember, it's sufficient to restore the higher order mutant defects. So how do you explain this? Do you think this is rather a dosage effect?
Yeah, this is a kind of a redundancy. When we knock out all these 3 genes, they show the phenotype growing randomly. If you knock out only 1 or 2, there's no clear gravitropic phenotype.
Okay.
Yeah.
Okay.
You put either one of them can rescue it. Yeah.
Yeah. Second, you mentioned this lazy also binds to lipid. Can you comment? Are there any specificity in terms of what kind of lipid species they bind, or it's a general? Yeah. Okay.
Yeah. So we actually only provided the in vitro assay to show that the lazy proteins can bind into some of the lipids, but we didn't go into the details in vivo to see the difference functions between this kind of different lipids. Yeah.
Okay. And one last question is for this MAP kinase phosphorylation, transient phosphorylation and trigger the lazy subcellular localization change. So do you have any ideas what kind of upstream signal to activate this MAP kinase signaling?
Yeah, this is a very nice question. Actually, we try to pursue the answer for this question. So we not yet get the answer, but we have some preliminary data to show that it is not the aminoplast sedimentation, because we check the phosphorylation of lazy proteins in the PGM1 mutant. In that mutant, the sedimentation is much lower. but the phosphorylation is the same. So it means that there are other gravity sensing mechanisms. We we look to figure out it in the future.
Okay, thank you.
Thank you for your question.
Hold on, can I ask a question?
Yeah, sure, Jiawei.
Yeah, very nice talk. So I have the first question is what's the downstream of lazy? So what's the biochemical function of this protein?
Yeah, this is a very nice question. So after the lazy was localized on the lower membrane, they will activate the oxygen redistribution, but there's a gap between lazy and those PIM proteins. We are trying to search for the gap because lazy have no biochemical activity. It looks like a scaffold protein. So there are some D6PK kinases that have been proposed to between them, but not yet get a connection between this protein and the lazy. So what we are doing, we're trying to use the lazy to search interaction proteins and we've already identified some kinase. Probably those kinases bring down together with the lazy and then activate the downstream PIM, those oxygen transport proteins. Yeah, that's our progress and what we think about. Yeah,
I see. So my second question is whether lazy is evolutionary conserved. So can you identify in the, for example, physical Mitrella?
Sure. We already get this kind of data. If we knock out the lazy in this plasma trailer, they show the random growth. So it means that in plasma trailer, the lazy is conserved. And now we also trying to look for the lazy sequence in the much earlier plants. Now the general conclusion is that the mouse lazy is there, but before that we could not find a very conserved ones. Yeah.
Okay, thank you.
Thank you for your questions. Yeah.
So hold on, I have a question. So it's a very interesting talk. So I'm wondering, do you what's your thing? What do you think about the upstream signal, about the gravity signal upstream of the lazy, or what's the initial event after the plants sense the gravity? So do you have any ideas?
Yeah, this is an important question, actually, similar to the question from the Libo, the first one. Actually, the general answer, we believe there's some other gravity sensing mechanism to initiate the lazy phosphorylation, but we have not yet get the answer. Yeah, probably we take some time to figure out it in the future. Yeah.
So do you think which of the methods or approach you will use to identify such signals?
Yeah, this is a good
just
question.
use the genetics or use other approach?
Yeah, we use several approaches. The first one is genetics, because we found the lazy proteins, the function is conserved in Phasmatrella, right? And Phasmatrella is very easy to do genetic screen. So we screen and obtain a lot of random growing mutant. Here the mutant is growing down, but we also have a lot of mutant growing randomly. Growing randomly means that the sensing or signal transduction was blocked, right? Yeah, probably in those mutants, hopefully we can identify these components involved in early sensing signals. And we also use some proteomics approaches, because the gravity sensing happened in specific cells, in columella cells, right? And in shooter endodermis. We're trying to do proteomics within these cells to check what kind of proteins probably have the response to gravity in these specific sensing cells. Yeah.
Okay, very good. Thank you.
Thank you, Xiaofeng.
Okay, it's time. Thank you, Haodong. Thank you again for a nice talk. Now
Okay.
let's move on
Thank you very
to
much.
the next. Okay, our third finalist for today's session is Dr. Li Sha-Shen, who comes from National University of Singapore, and she will deliver an interesting talk on N6 method Indonesia RNA modification in plant development. Nisha.
Hello. Can you see my screen and hear me?
Yes. Yes.
Yeah, great.
What's
Okay. Yeah. Thank you, Ron, for the introduction. And hello, everyone. I would like to thank the organizers and the the scientific community for this great opportunity to share the research progress in my lab. So the topic of my presentation is N6-methyladenosine iron modifications in plants. So the chemical modifications to RNAs are collectively termed the epitranscriptome. It refers to the functional relevant changes to the transcriptome that do not involve a change in the RNA sequence. So currently, there are over 170 different chemical modifications to RNAs has been discovered. Most of them are identified in the non-coding RNAs with high abundance, such as the tRNAs and the RNAs. So until recently, the technical advances in detecting and mapping RNA modifications in low-abundant RNAs have allowed the studies of RNA modifications in messenger RNAs. So in plants, mRNAs are known to have several RNA modifications, including both the cap modification and the internal modifications. So among the internal modifications, M6A is the most prevalent. M6A is short for the N6-methyladenosine, and it refers to the methylation of adenosine at the N6 position. So it is a conserved modification found in many different species. And M6A is essential for many organisms, and it has been shown to affect various aspects of iron metabolism from transcription to decay. So M6A is a reversible modification, and the M6A dynamics requires the evolutionary conserved M6A effectors, including the writers for M6A deposition, the erasers for M6A removal, and the M6A readers for recognition and interpretation. So the deposition of M6A can be catalyzed by two different groups of M6A writers. The first group is the M6A writer complex, and the second group is the phenol-1 orthologs. So in Arabidopsis, the M6A writer complex contains two methyltransferases, MTA and MTB. So according to the animal model, MTA and MTB may form the MAC subcomplex that constitutes the catalytic core. So the writer complex also contains several accessory subunits, including FIP37, VRR, HiCi, and HIZ2. So these accessory subunits form the MACOM subcomplex that may regulate the activities of MTA and MTB. So the second M6A writer phenol-1 has also been identified in Arabidopsis, which I will discuss later. So M6A is a reversible modification and can be removed by the ALKBH proteins. After deposition, M6A is recognized by several reader proteins, such as the proteins in the YTH domain protein family, which could regulate iron stability, alternative pollination, and translation. So there will be two parts of my presentation. So in the first part, I will mainly discuss the M6A deposition by these two different groups of M6A writers. And in the second part, I will introduce our recent work on the M6A reader. So let's first take a look at how M6A deposition is mediated by these two groups of M6A writers. So since the complete loss function mutant of the major components of M6A writer complex genes leads to embryolysis, we use the artificial microRNAs to knock down the expression of MTA and VRR, and we use the embryo-specific promoters of ABI3 or LEC1 to complement the embryolysis phenotype of MTB and FIP37. So the HiCi mutants was generated by CRISPR-Cas9. So the resulting MTB and the FIP37 mutants show a strikingly large shoot apical meristem phenotype and serrated leaves, as you can see from the enlarged photos. MTA and the VRR knockdown plants shows a delayed growth, abnormal leaf shape and margins at the early stage, and they will also develop meristem-like tissues at a later developmental stage. In contrast, the high-Chi mutants do not have obvious growth defects, so the severity of the phenotypes are consistent with the reduction of total M6A levels. In the mutants of MTA, MTB, FIP37, and VRR, the total M6A level were greatly reduced, but in high-Chi mutants, the M6A level is only slightly reduced. So these observations suggest that the MTA, MTB, VRR, FIP37 are essential for shoot meristem development and M6A methylation. So the first question we asked was, how does M6A control shoot meristem development? We have found that in wild-type plants, these M6A writers mediate the M6A modifications of USO and STM transcript, which are two essential regulators of meristem development. So M6A modifications on these USO and STM transcripts promote their messenger RNA decay to confine their expression domains in their confined expression domains and resulting in normal meristem development. So when M6A methylation is abolished in these M6A writer mutants, USO and STM transcripts are stabilized, resulting in enlarged meristem phenotype. So since all these four M6A writers are necessary for meristem development and M6A methylation, so the second question we asked was how these writers affect each other to fulfill the function of the M6A writer complex as a whole. So we have found that these writers, they do not affect each other at the transcriptional level, but they affect each other at the protein level. So first, MTA and MTB affect each other's protein accumulation, and MTA also affect the subcellular localization of MTB in the nucleus. Second, the accessory subunits FIP37 and VRR affect the protein stability of MTA and MTB. And third, the VRR influence the protein accumulation of FIP37 and high-Chi. So their counterparts in mammalian cells also affect each other at the post-translational levels, but they mainly through affecting the protein localizations. So we propose that this unique functional interdependence at the post-translational levels among the individual components in the M6A writer complex is essential for M6A methylation and its function in plant development, such as the shoot meristem development. At the molecular level, we determined the M6A site dependent on these individual M6A writers and found both common and unique target of these M6A writers, so supporting their functional interdependence, but also pointing to their specific functions in M6A methylation. So these M6A target sites are mainly located in the 3' UTRs of transcripts, and M6A mainly affect gene expression and poly-A site selection. But during the course of study of these M6A writers, we found that the disruption of these genes, they result in a greatly reduced but not totally abolished M6A levels, and they mainly affect the M6A methylation in the 3' UTR and the sites near the stop codon, but not the sites in the coding regions. So the next question we asked is whether there is other M6A methyltransferase in Arabidopsis. So a good candidate is Fiona-1, as it is an ortholog of the human metal-16. So the human metal-16 installs M6A on diverse iron molecules, such as the U6 small nuclei, non-coding and non-coding RNAs, and the pre-mRNAs of several protein coding transcripts. So Fiona-1 is the only Arabidopsis ortholog of human metal-16, and it contains a methyltransferase domain at its N-terminus. So we isolated two Fiona-1 mutants. Both Fiona-1 mutants shows a very early flowering phenotype under both long day and short day conditions. dot plot using the anti-M6A antibody revealed that the total M6A level is reduced in Fiona-1 mutants. Now, further quantification shows that the M6A levels of both total RNA and messenger RNAs were reduced by around 10%. So these results suggest that Fiona-1 is involved in M6A methylation. So Fiona-1 contains the NPPYF catalytic core in its methyltransferase domain. Fiona-1 possesses the in vitro M6A methyltransferase activity, but when this K-NPPY motif is mutated, the mutated Fiona-1 lost its methyltransferase activity. And in planta, this mutated Fiona-1 failed to rescue the early flowering phenotype of Fiona-1 mutants, suggesting that the M6A methyltransferase activity is essential for Fiona-1's function in flowering time regulation. So to determine the methylation targets of Fiona-1, we determined the changes M6A in Fiona-1 mutants using the nanopore direct RNA sequencing. So we calculated the differential modification rates that represents the difference between the modification rates in wild type and Fiona-1. So in total, we have identified over 3,000 high confidence Fiona-1 dependent M6A sites. So these sites are located in around 2,000 protein coding genes, and we also identified a consensus motif, YHAGA, for Fiona-1 methylation, which is clearly different from the motif for the M6A Reta complex. Gene ontology analysis revealed that the hypermethylated genes carrying at least one hypomethylated M6A site could regulate multiple biological processes. Interestingly, the genes involved in the plant reproductive process and the rhizomatic process were enriched, which could be associated with the early flowering phenotype of Fiona-1 mutants. So in line with the early flowering phenotype of Fiona-1 mutants, the hypomethylated genes include the famous floral pathway integrator gene SOC1, and SOC1 contains a hypomethylated site in its 5' UTR region. We then analyzed the SOC1 expression in Fiona-1 mutants to examine whether this hypomethylated M6A site affect SOC1 expression. So SOC1 expression was constantly upregulated in Fiona-1 mutants, which is consistent with its early flowering phenotype. So Fiona-1 is directly associated with SOC1 transcripts, suggesting that Fiona-1 directly mediates the M6A methylation on the SOC1 transcripts to modulate its expression. And genetically, the late flowering SOC1 mutants suppressed the early flowering phenotype of Fiona-1, supporting that Fiona-1 functions through SOC1 to prevent the early flowering. So that's we propose a model depicting the function of Fiona-1 in M6A methylation and the flowering time control. We identified Fiona-1 as a new M6A methyltransferase that is involved in depositing M6A methylation, mainly in the coding regions of a subset of protein coding transcripts. Fiona-1 directly mediates the M6A methylation on SOC1 to regulate its expression, and it also methylates other flowering time regulators to indirectly affect SOC1 expression. Interestingly, Fiona-1 does not regulate expression levels of other M6A Reta genes and the eraser gene, and it does not physically interact with the components of the M6A Reta complex, implying that these two groups of M6A Retas may function in parallel for M6A deposition in Arabidopsis. So after M6A is deposited by the Retas, it could be recognized by different M6A readers for various downstream events. So in the second part of my talk, I will share with you how M6A recognition by M6A readers affect mRNA metabolism. So Arabidopsis has 13 YTH domain containing proteins. which can directly bind to M6C through the highly conserved YTH domain. And these proteins are named ECT1-12 and CPSF30L in Arabidopsis. So based on our observation, most single loss function mutant of this YTH domain genes do not greatly affect plant development. So we then focused on their function in stress response. So among these 13 YTH domain genes, we found that the expression of ECT8 was greatly upregulated after ABA treatment. So we then isolated the 2 mutants of ECT8. Both ECT8 mutants do not show obvious defect under normal conditions, but showed an ABA hypersensitive phenotype, including the decreased cotyledon graining rates compared to wild type under the ABA treatment. And the ABA hypersensitive phenotype can be rescued by the genomic construct of ECT8. So in addition to the conserved YTH domain, ECT8 were also predicted to have 3 intrinsically disordered regions, IDRs. So IDRs are often found in proteins with the liquid-liquid phase separation properties. Under the normal conditions, ECT8 is relatively evenly distributed in the cytoplasm, but ABA treatment induced the aggregation of ECT8 to form granule-like structures in the cytoplasm. The ECT8 condensates could quickly fuse with each other to form one granule in the cytoplasm. And the fluorescence recovery after photobleaching showed that the ECT8 was able to rapidly redistribute from the unbleached areas to the bleached areas. So this observation suggests a liquid-like state of the ECT8 condensates, and the formation of condensates is induced by ABA treatment. Since the ECT8 possesses the YTH domain that could bind to M6A, we then proceeded to examine whether the M6A binding capacity of ECT8 is required for the condensate formation. So we mutated the two key residues in the hydrophobic aromatic cage essential for M6A binding. In contrast to the ECT8 native protein, this mutated form of ECT8 lost its M6A binding capacity. And interestingly, we found that when this ECT8 is mutated, it can, under the ABA treatment, it can no longer form condensates in the cytoplasm. And also the mutated ECT8 could not rescue the ABA hypersensitive phenotype of ECT8 mutants, so suggesting that the M6A binding capacity of ECT8 is essential for its phase separation ability and its function in ABA signaling. So we then identified the ECT interactome to understand the properties of ECT8 condensates. So under the ABA treatment, the ECT8 interactome was enriched with several known components of stress granules, including the RBP47, which is a marker of the stress granules. So the stress granules are dynamic and transient cytoplasmic condensates containing proteins, mRNAs, and metabolites under stress conditions in eukaryotic cells. So indeed, we observed the co-localization of ECT8 condensates with the RBP47B upon ABA treatment, suggesting that the ABA-induced ECT condensates are localized in stress granules. Since we have known that the ABA-induced condensates are localized in stress granules, and the formation of condensates depends on the M6A binding capacity of ECT8, we then asked whether there is any link between the stress granules and M6A modified mRNAs bound by ECT8. So in mRNAs isolated from stress granules, the M6A level was greatly reduced in ECT8 mutants, while in the total cellular RNAs, M6A level in ECT8 is comparable to wild type. So this suggests that the ECT8 may recruit the M6A modified messenger RNAs into stress granules after ABA treatment. So we then performed the nanopore direct R sequencing using the mRNAs isolated from the stress granules, and we identified over 6,000 hypomethylated M6A sites in ECT8 mutants, while only around 1,000 hypomethylated M6A sites. So this supports that ECT8 selectively recruits the M6A modified messenger RNAs to stress granules. So among the hypomethylated transcripts identified in stress granules in ECT8 mutants, we found PYL7, which is an ABA receptor. So PYL7 is modified by M6A methylation, and in ECT8 mutant, M6A levels in PYL7 in stress granules is reduced, and its accumulation in stress granule is also reduced. And we found that the PYL7 messenger RNA showed a co-localization with ECT8 protein in stress granules on the ABA treatment. And interestingly, the total levels of PYL7 was much higher in ECT8 mutants compared to the wild type on the ABA treatment. And genetically, the PYL7 mutants largely suppressed the ABA hypersensitive phenotype of ECT8, suggesting that the ECT8 phenotype is attributed to the increased accumulation of PYL7 protein. So we thus proposed a model summarizing the function in M6A, the function of ECT8 in M6A recognition and the stress response. So in the presence of ECT8 in wild type, ABA upregulates the expression of ECT8 and triggers its liquid-liquid phase separation to form stress granules. And during this process, ECT8 selectively sequesters the M6A modified transcripts, including PYL7, for temporary mRNA storage. And this restricts the amount of PYL7 messenger RNA for translation, thus conferring a negative feedback regulation of PYL7 protein levels and ABA signaling. So here shows a summary of our work in M6A modification in recent years. So M6A methylation requires two groups of M6A retters. So the first group is the M6A retter complex. We have shown their function in short apical meristem development. And there is self-regulatory network among the individual components, and there are common and unique targets. So we have also identified another M6A retter phenol1, which acts in parallel for M6A methylation as function in flowering time regulation. So we have also reviewed the phase separation of M6A reader in mediating ABA response. So M6A is a well-conserved RNA modification in many plant species. So we have shown that the M6A retter in rice in controlling the male fertility. And recently, we have also mapped M6A methylation and studied its role in the regulation of whole genome duplicated genes in one of the most important vegetable crop, the lettuce. Hope that I can share with you more progress in the future. So I would like to thank to the postdocs involved in these projects, and also thank to my collaborators, especially Professor Yu Hao, who is also my former supervisor, and also thank the various funding agencies for funding this project. And thank you all for your attention, and I'm happy to take questions.
Thank you, Lisa. Thank you. Libu?
Yeah, Lisa, very interesting work. So I have a question for your ECT8 LLPS promoted by ABA cycling. So you propose this M6. So basically, the M6A mRNA is required for ECT8 LLPS. Right, in your model?
Yes.
So this is based on the mutation lost LLPS effect. Then do you, if you add M6A modified mRNA, do you see this promotes ECT8 LLPS?
Yes, I didn't show the result here. We have observed the ECT8 condensate formation in one of the M6A retinal mutants, the 537, which lost M6A, and we found that in this M6A mutant background, the ECT8 phase separation is largely compromised and can no longer form condensates in the cytoplasm under the ABA treatment.
Okay. So how the stability of ECT8 that M6A mRNA binding site mutation, is the protein still stable?
Yes, yes.
Okay.
Yeah.
Okay. And for other ECT family members also have this distinct IDR regions. You think this is a universal mechanism for other ECT members?
Yes, actually, most of the ECT proteins, they have this, they have IDR regions, and there are other studies showing the phase separation of ECT8 proteins is essential for their function in the in interpreting M6A. So I think it is probably is the phase separation ability of the ECT family proteins is a common mechanism for that is essential for M6A reading reading functions. Yeah.
And another thing is also for most of ECT are transcriptionally regulated. Here you propose this abiotic stress ABA response mainly through transcription regulation, right? So the other member of the ECT, you also observed neurodevelopmental or other biotic stress dependent transcription regulation?
Yeah, it's a very interesting question. I think under our observation, the ABA factor ECT8 is probably not observed in other ECT members, but we do not exclude the possibility that maybe other stress or developmental signals will affect the expression levels of ECT family proteins. Yeah.
Okay. Yeah. Thank you.
Thank you.
Thomas, you have questions?
Yeah, so I have a question that relates actually to the last question of Libo. So you use the ABA to form stress granules. So of course, stress granules are also formed, for example, by heat stress. So I'm wondering, do you also see ECT8, for example, in stress granules that are formed by heat stress or salt stress or any other kind of stress?
Yes, the answer is yes. Actually, we did several other stress treatments. For example, the mannitol and sodium chloride, the high salt, and as well as heat treatment. They all can induce the phase separation of ECT8 to form condensates in the cytoplasm.
So it's a very conserved mechanism that you found.
Yes.
Yeah, really great work. Thanks.
Thank you.
So these are a very nice talk. So I have a question. So you talk about the FIO1 mediated M6A methylation control flowering, but ECT8 recognize M6A modification at the PYL7 involved in the ABA signaling. I'm wondering, so different methylation, MCCD methylation, how they recognize different targets, and how the readers, how the also target different gene or target message RNA. So do you have any ideas about the,
Okay, yes, thank you. I think it's a very interesting question that we are actually we are looking into the answers about the specificities of the M6 writers or readers. So in So based on the current studies, I think the specificities of the M60 retters, they may achieve this through their interactions with different many other different proteins, such as some RNA binding proteins that may mediate the specificities of M60 retters. For example, in rats, we have found that there is a RNA binding protein that interact with the OS-57, which will confer its specificity in regulating some targets during the male reproductive organ development. And also for the M60 readers, I think probably there is some similar mechanism, but we don't know now. And this is a question that we are always thinking about, and we try to answer these questions in the future.
Okay, thank you. So you mean that the RNA binding protein will play a very important role in the M60 mediated processes like the transcription factors, binding the DNA.
Yes.
Okay.
Yes.
Thank you.
Okay.
Thank you.
Thank you, Lisa. Due to time limit, we have to move on to the next speaker.
Okay. Thank you.
Thank you. Our last finalist for today's session is Goldsday DeMeyer. She comes from California Institute of Technology, USA, and will talk about accelerating plant biology discovery with an improved genetic engineering toolkit. Please join me to welcome Goldsday.
Okay, thank you, Dr. Chen, for that introduction. I hope my slides are visible and everyone can hear me well. All good?
No, it's black now. Okay, now okay.
Okay, it's sharing?
Yes.
Okay, perfect. Okay. Yeah, thank you for the introduction. I'm really excited to be here, and I'm really honored to be selected as one of the finalists for the Rising Stars in Plant Sciences contest this year, along with 7 other amazing scientists. I'm excited today to share some stories from my research that is on developing nano and biotechnologies for improving both the plant and the planet health. So I'm going to start with our motivation. And the main question that has been motivating my work so far is to achieve food security for a growing global population living under changing climate using more sustainable approaches. So currently, how we do agriculture ends up contributing to climate change substantially through greenhouse gas emissions and let me get my pointer here. Yeah, through greenhouse gas emissions and excess usage of chemical fertilizers and pesticides, and many practices that we do to manage our soils and crops. And in return, climate change, as we all know, causes increases in temperature, extreme availability or unavailability of water, along with a lot of new and drastic diseases targeting plants. Then these effects of climate change decrease crop yields. To increase our agricultural productivity, we then apply more harmful approaches, and this cycle has been propagating in a negative way for quite some decades, right, until so far. So my lab and many other labs in the field think that plant genetic engineering is a promising and sustainable approach to boost crop yields and render them resistant to climate change as well as new diseases without further harming the environment. However, there are several bottlenecks preventing plant genetic engineering from reaching its full potential. So first and foremost, our tools are highly limited in terms of biomolecule delivery into plants and for their genetic transformation, as well as CRISPR genome engineering still faces some unique challenges in plant systems. Given these tool limitations, as well as large redundant plant genomes, we still have big knowledge gaps in understanding gene function and regulation, as well as important developmental and environmental processes that contribute to plant health and yield. Last but not least, we also face major challenges in translating these genetically engineered crops from lab to field. And some of these challenges are because of societal and regulatory barriers of genetically engineered plants, but some of them are also technical challenges, as we don't know the interactions of these engineered plants and sometimes even wild-type plants with the environment and the organisms that are living in those environments. And this could prevent the beneficial traits that we demonstrate under optimal lab conditions to be effective in field conditions. So my lab's research has been aiming to address these challenges and accelerate the plant biology discoveries through an improved genetic engineering toolkit. And we focus our efforts under three main directions. In the nanotechnology area, we have been developing novel nanomaterial platforms for biomolecule delivery and plant genetic transformation that is improved and available in broader plant species. Using these improved delivery approaches, we have also been optimizing gene expression and gene editing machinery in plants to enable more spatiotemporal control. And we have been also focusing on specifically improving the targeted gene insertion capabilities in plants. And lastly, we have been using these improved nano and biotechnologies to answer basic and applied research questions in rhizosphere engineering, where we aim to elucidate the gene regulatory networks controlling plant nutrient use efficiency. We're also trying to understand these dynamics and complex plant microbe interactions and monitor some of the important soil microbial activity for productive and sustainable growth of plants. So today, in this relatively short talk, I'm going to give you one or two example projects from each of these areas without going into too much detail. And I'm going to start talking about this gene delivery approach that we have developed using carbon nanomaterials. So what's been motivating our nanomaterial mediated delivery projects is these plant transformation challenges that we face, where direct transformation of plant germline cells is only possible in the model plant Arabidopsis thaliana, and we do this through Agrobacterium tumefaciens-mediated flower dip as shown here. So even though this has been very instrumental in enabling a lot of plant biology discoveries so far, this approach, as I mentioned, only works in this model plant Arabidopsis, and for every other plant and crop species of interest, we need to transform tissues or cells ex vivo and then go through this regeneration process in tissue culture to create the engineered plants. So this approach is limited to only certain plant species and even within species to only certain cultivars, and it needs a very extensive optimization process. So even in tractable species that we can engineer, this process can take very long time and require a lot of resources. And the final plant ends up containing transgenes such as Cas9. In most cases, we need to remove them, which adds even further labor and time to these processes. So given these very critical limitations of plant transformation, I have been focusing since my PhD in UC Berkeley in Marquita Landry's lab to develop nanomaterials that can address some of these challenges and deliver biomolecules into plants for transient DNA expression, as well as silencing and CRISPR-Cas9 genome editing. Since all of this work is already published at this point and can be accessed in these publications you see here, I'm only going to give a very brief summary of this DNA delivery work as it currently relates to the efforts in my own lab for plant transformation. So here I use these modified single-walled carbon nanotubes to deliver plasmid DNA into the leaves of mature plants, here shown for Nicotiana bentamiana, to express reporter genes like GFP. And we were really excited to see that this platform is amenable to a lot of different dicot and monocot species for transient expression in their leaves. Here, for example, other than Nicotiana bentamiana, none of these other species had good transient leaf transformation protocols, as well as some of the species here, such as sorghum and many of its cultivars, did not have any way of being able to do genetic manipulation before. So we were really encouraged and excited about the potential of the broad species compatibility of these nanomaterial mediated delivery methods. Another advantage of this nanotechnology is that the DNA gets expressed transiently. As you can see, that the protein and mRNA disappears after 7 or 10 days without causing any insertion of the delivered plasmid or its pieces into the plant genomic DNA, which we know this is not the case in, for example, agrobacterium, the common way of doing genetic engineering in plants, where you get delivered genes randomly insert somewhere in the plant genome. So we wanted to take advantage of this fully transient expression platform to express CRISPR machinery, such as Cas9 and guide RNA, transiently to cause permanent gene edits in the leaves of Nicotiana bentamiana. Here you'll see in these leaves without selection, somatic gene editing, we have comparable efficiencies to that of agrobacterium. However, the advantage is that in our case, our approach could be used in the other species, as I've shown in the leaves of other species, and that in our approach, this Cas9 and guide RNA did not get inserted into the plant genome, which we know can cause problems and needs to be removed most of the times in the next generation of plants. So in summary here, we have developed a new transient transformation resource that can be applied to a wide species range, as well as it's non-integrating, easy and quick to perform, and it's biocompatible. I'm not showing any of the toxicity results, but they're in our paper here if you want to check those out as well. So even though this transient transformation has many impactful uses in basic and applied plant research, and we are following up some of those applications in my own lab, we have also been really extremely interested in being able to use these nanomaterials for stable and heritable plant genetic engineering. So when we consider which tissues to target for this purpose, we realized that tissues like callus, embryos, or cells like protoplasts are easier to deliver cargoes inside, but their regeneration is laborious and time-consuming and could be difficult. Whereas it would be easier to recover the next generation of edited plants if we could engineer pollen, flowers, or seeds directly, but these tissues are typically very hard to access and deliver cargoes in. So we were able to deliver our carbon nanotubes to callus and embryo, but as I mentioned, we also want to develop a tissue culture-free and an easy transformation approach without going through this entire regeneration process, so we didn't want to pursue these type of tissues. We tried delivering carbon nanotubes into pollen. Here you're seeing the nanoparticles inside this pollen. This is a cross-section. But what we saw is that the pollen easily loses viability after getting exposed to nanomaterials. So this wasn't a viable option either. So in my lab, we decided to target plant shoot apical meristems. As shown here, they contain a pool of stem cells with a capability to naturally give rise to all above-ground plant tissues, which include germ cells, flowers, and eventually seeds. However, when we tried delivering these carbon nanotube platform that I showed you into these meristems, they were unable to enter this tissue. So we investigated other types of carbon nanomaterials and hypothesized that these carbon dots, which are substantially smaller and spherical particles, may be more advantageous for meristem entry, because we suspected that the carbon nanotubes, having properties and dimensions very similar to plant viruses, may be recognized and prevented from entering into these stem cells. So I'm going to skip the synthesis and characterization of these carbon dots, but we tested them in plant shoot apical meristems. We work with dicot plants, and in the dicots, the shoot apical meristem is at the growing tip of these plants. Here you're looking at the shoot apical meristem. And if you look at a cross-section of this, there is this layer of cells, stem cells, in the L2 layer that specifically generate germ cells. So we wanted to target our nanoparticles into the stem cells in the L2 layer. So we were really excited to see these preliminary results where we dip shoot apical meristem into a carbon dot solution and vacuum it briefly. These nanoparticles are able to enter the cells in the shoot apical meristem, and not only the cells, but it seems like they're also effectively able to enter the nuclei of these cells. So to our knowledge, this was the first time delivery into meristems and stem cells in intact plants, as you can see here. And when we took a cross-section of these shoot apical meristem tissues, we also saw the carbon dots in the L2 layers as we wanted. So we spent quite some time optimizing this delivery system, such as the properties of vacuum, dissecting the outer tissues a little bit to expose the shoot apical meristem area. So we wanted to see if then this delivery could be used to deliver agrobacteria as well, or if it's unique to carbon dots. So when we use the same treatment and deliver agrobacteria to express GFP in this tissue, we don't see it working. So agrobacteria is not able to reach and express in these tissues, showing that the carbon dots and nanomaterials have unique properties to be able to access these previously inaccessible tissues and cells. So what we are doing right now is extending this delivery approach to transiently express CRISPR cargos in shoot apical meristems. And if we could edit the cells, the stem cells in the shoot apical meristem, then these stem cells can generate edited germ cells, flowers, and seeds, which we can easily collect in a couple of weeks without needing to regenerate in tissue culture. And that will massively improve the way we perform stable plant transformations. And given these nanomaterials could potentially work in broad species range, currently we are testing this both in Nicotiana benthamiana and in tomato, but we also want to translate it to other crops going forward as well. So now I'm going to shift gears and talk a little bit about our efforts on increasing the targeted gene insertion efficiency in plants without relying on double-stranded breaks or HDR. So we all know that targeted DNA insertion is an important capability, both in fundamental biology and applied science, to, for example, give plants improved traits, as well as elucidate genotype-phenotype relationship. The common way of achieving targeted gene insertion is to use Cas9 to create double-stranded breaks and provide a donor template, a DNA that we want inserted during double-stranded break repair using homology-directed repair in plants. But double-stranded breaks can cause insertion and deletions in cells, genome rearrangement, and potential cellular toxicity. And HDR is infrequent DNA repair pathway, especially in some of the cell types that we're interested in, making this targeted gene insertion to have low efficiency, typically around single-digit efficiencies. So recently, a newer CRISPR system was discovered in bacteria called CRISPR-associated transposons, where transposons and the transposase complex naturally associate with Cas effectors like Cascade or Cas12K. And this causes RNA-guided targeted transposition into the genome of the species. So this was discovered in bacteria. Vibrio cholerae is one of them. It's a type IF Cas system, and Cytomania hoffmanni is another one that has a type VK CRISPR-associated transposon systems. So after its initial discovery, a lot of people have used this in a lot of different bacteria species initially, where you could see almost 100% targeted gene insertion after selection in bacteria cells, and in human cells as well. You could use this Cas system, though with lower efficiencies. And this paper recently showed that there is another type IF system from another bacteria called Pseudoalteromonas that you could achieve higher gene insertion in eukaryotic organisms. And more recently, there has been an application of a synthetic Cas system in plants where researchers merged Cas9 with plant transposons. And you could see around 10 to 15% targeted gene insertion efficiency that is typically higher than a regular HDR, demonstrating the promise of these systems. So in my lab, we have been working on translating the native bacterial Cas systems into plants for targeted gene insertion. The advantages are that you wouldn't create a double-stranded break, you wouldn't need homologous arms to do the insertion. The PAM is more flexible in this approach, and it has the capacity, given bacteria results, to insert large cargos at a high efficiency, which we would need to show in plants. So we've been working with both systems, also from Vibrio cholerae, Pseudoalteromonas, but today I'm only going to be showing the Vibrio cholerae results, which we shorten as VCH-Cast. So this project has definitely been kind of a large undertaking, just even to express all the proteins that we needed to express. So it took a lot of optimization of circuit architecture, NLS type, location, promoter strength to even be able to express these proteins in plants at high level. So here I'm showing finally that we are able to express all of these proteins. Here you're looking at the YFP-TEC proteins, as well as we use this hybrid luminescence complementation method to detect them at higher throughput manner in a plate reader. So you can see even after all that optimization, the transposases have lower expression in plants. Plants really don't like expressing these type of proteins, but we were recently able to overcome this by putting these transposases under Gemini virus replicant vectors to increase their expression efficiency. So now that we can express all of these components, we moved on to the targeted insertion experiments using CAST. And in the early experiments, we used agrobacteria to express these proteins that will cause random insertion. So we needed to get clever with the design of the DNA so that we could distinguish the random insertion from the insertion caused by casts. So what we did is we inserted a coding sequence of YFP that doesn't have a promoter right after a 35S promoter, either in a plasmid in protoplast or in the chromosomal chromosome integration of transgenic Nicotiana bentamiana plants that contain the 35S promoter. So in either case, the YFP fluorescence will only be visible if it's inserted through targeted insertion and not through random insertion. And this is exactly what the results show, that we can cause targeted insertion using casts in plants. And we also confirm this by sequencing, where we get the correct sequence, expected insertion location, as well as the hallmark of CAST system, which is 5 base pair target site duplication around the site of the donors. So we are confident that the insertion happens through the CAST-mediated approach, and we are currently working on increasing the cargo size and insertion efficiency, as well as delivering these with our nanoparticles instead of agrobacteria, so we can remove the random insertion altogether. And we are going to hopefully use these to create stable transgenic lines in addition to our transient leaf experiments as well. Okay, and in the last minute I have, I'm going to very briefly talk about some of our efforts to increase plant nutrient use efficiency sustainably. So as we know, nitrogen and phosphorus are vital nutrients for crop yields, but their synthesis, procurement, and excess use has been causing many negative consequences, such as the eutrophication, atmospheric deposition of ammonia, emissions of nitrous oxide, which is a very potent greenhouse gas, and soil degradation. So our research focuses on 2 approaches for more sustainable improvement of crop yields. In one area, we want to take advantage of plant beneficial rhizobacteria for plant nutrient uptake. So by modifying the plant root exudates, we can alter the composition and function of the microbiome, and plants can already do this to a certain extent, but over many decades of domestication, as we prioritize other traits, these beneficial root microbiomes got dysbiosis, and this situation got worsened because we always had excess nutrients in the soil because of the chemical fertilizers, so plants lost ability to enrich these type of microbes. So in our approach, say there's a nitrogen deprivation, the plants will synthesize and exude a specific metabolite that can specifically recruit and enrich nitrogen-fixing bacteria. And similarly, when there is low phosphorus, they can recruit and enrich phosphate-solubilizing bacteria by secreting a specific chemical that is preferentially used by these type of bacteria. So we have already identified what these specific metabolites are to enrich these nutrient beneficial bacteria, and we showed that we can enrich them over native soil microbiome when we exogenously add these molecules. So what we are doing currently, we are trying to have plants synthesize and secrete these molecules using synthetic biology approaches, and that we are testing the growth benefit of enriched microbiome under nutrient limiting conditions. In another research area, we are directly engineering plant genes and their regulation to improve plant nitrogen and phosphorus efficiency. For instance, in this project that started during my postdoc in Shubham Brady's lab at UC Davis, in collaboration with the Patron Lab, we first elucidated some of the complex gene regulatory networks that control nitrogen use efficiency both in Arabidopsis and tomato, and then use these predictive network models to rationally engineer tomato for better nitrogen responsiveness. And similarly, we worked with phosphorus response in domesticated tomato, Solanum lycopersicum, versus this wild tomato species, Solanum pennellii, and we showed that pennellii was insensitive to phosphorus deprivation because it had a constitutive response even under phosphorus sufficiency. And we realized that this was through an activated bristle steroid signaling through the transcription factor BZR1 that depends on zinc accumulation, and we could cause the same phenotype in the domesticated tomato and make it insensitive to phosphorus deprivation through CRISPR gene editing. So today I've shown you some of our efforts to accelerate plant biology discoveries using an improved genetic engineering toolkit, such as improved and stable transformations, as well as high efficiency targeted gene insertion in plants, and improving plant nutrient efficiency using sustainable approaches. With that, I would like to thank my lab at Caltech, as well as our funding sources, and my postdoc lab, Brady Lab at UC Davis, and PhD lab, Lander Lab at UC Berkeley. Thank you all for listening, and I'm happy to take questions now.
Thank you. Any questions from committee members? People.
Yeah, hi, Gozi. Really interesting perspective project as well. So for the carbon nano delivery. So you think for different plant species, the pollen, the feature of pollen code also make this unique in the way they kind of relatively easy adopt about certain pollen. So do you think there is a plant species dependent feature in terms of the efficiency to uptake these carbon dots?
Yeah, that's a great question. We have done very minimal work with pollen towards the end of my PhD, but I agree that I think species will have different properties and that could be very important in pollen delivery. So we specifically worked with pearl millet pollen in that project, and we were able to put nanoparticles in them, but they wouldn't germinate their pollen tube after getting exposed to carbon nanotubes. So I think maybe we needed to optimize either the treatment or the concentrations of carbon nanotubes. We didn't pursue that work further because that was the end of my PhD, but I think there could be a lot of promise. For example, maize pollen could be another good target. If we could put these particles in without affecting their fertility, it could be a really nice way to then artificially pollinate with the engineered pollen. Yeah, so we need to do more. Yeah.
Okay. And for the mini transposon assisted insertion, so do you see that the let's say YFP is inheritable for the next generation? So far, you saw transgenize it works.
Yeah, so you only did so far the transient leaf experiments, and we didn't like try to regenerate the new plants from that. We are currently doing Arabidopsis flower dip with this system, and we just collected our seeds, so we will need to see. So technically, it should be heritable because it does insert into the chromosome. So unless it gets silenced, because it could get silenced potentially, but the good thing is that if we deliver them with nanoparticles, we won't be inserting the transposases and Cas, so that's likelier to be more effective in the long term, because the problem with transposases, plants don't like them, so they try to silence them a lot, or they try not to even express them. So I think if we could just transiently express the transposases for a very short time to insert it into the chromosome, then hopefully what's inserted into the chromosome could stay in there and express, because it depends on the cargo, like YFP typically seems to be stable. But yeah, but we are currently doing it in Arabidopsis, and hopefully I'll have more results on that soon.
Thank you. Thomas?
Yeah, maybe, hello. Maybe one question regarding the delivery of the carbon dots. So, of course, ideally, also in the future on a field application, you may fly with drones over fields and spray those dots that contain RNA or other molecules. So I'm wondering, so how is it taken up by the cells?
Yeah,
Because
that's a great
cells, of course, have a thick cuticle, and
So we apply.
spray from the surface. So, and you have, for example, stomata and many plant species on the bottom of the leaves.
Yeah, right. Yeah, all great questions. So with the leaves, we do like a syringe infiltration, like agrobacteria from the bottom side of the leaves, and then it probably enters through stomata, and then these particles are able to cross through the cell wall and cell membrane. We are still working on kind of trying to understand how that happens, but it seems like they are very stiff and small, so they could either pass through the pores of the cell wall or puncture their like own holes as they go through the cell wall. But if you think of a more scalable application, obviously you shouldn't probably go and inject like each leaf one by one with a syringe. That wouldn't be very scalable, right? So a couple of things that we've been doing is that, again, not so relevant to field applications, but we can do vacuum. So you could put the entire plant into a vacuum chamber, dip it in a solution and apply vacuum, and the particles are able to then enter tissues as you vacuum it, right? When the water goes out of the leaf, the new solution goes in. But again, vacuum is probably not going to be so translatable to field applications. So what we've been doing for that is that if you just put a drop on a leaf or something, it doesn't go in. There's no pressure pushing it in. So you need to have surfactants like Silvet. So we show that if you have Silvet in the solution, it is able to penetrate into the leaf tissue. So I guess when you are thinking of a spray application, you could consider putting a Silvet in the solution, but we still need to do a bit more work on that to enable like an efficient entry into these tissues that wouldn't require either syringe or a vacuum type of treatment.
Thank you.
Got the next talk. So I have a question about how you prepare the carbon nano dots or nano materials chemically. So can normal, okay.
Did you say how do we produce?
Yeah.
Mm-hmm. Yeah, so that was another reason to switch from carbon nanotubes to carbon dots, because carbon dots are very easy to synthesize. So we mix glycerol as a carbon source and polyethylenimine, which is a positively charged polymer. So you mix two of those ingredients and you heat it in the microwave, and then that kind of creates these nanoparticles. Because it's through microwave, it's not very controlled synthesis, but we like it because it's like very scalable and very easy. Everyone can generate it. But then after that, you need to purify them, because we only want to keep the very small particles. The large ones are not able to enter plant cells. So we pass them through these spin filters. People used to sometimes like purify proteins and concentrate proteins. So if you have a correct spin filter with the desired molecular weight cutoff, you could collect only the small particles. So that's kind of the synthesis, mixing two of those very simple ingredients, heating and purifying for size.
So you mean the biological students, biological scientists just follow the protocol can also produce the carbon dots?
Yes,
it's
what
very
we think.
easy?
Yeah, because we had problems with carbon nanotubes. They are harder to synthesize, and plant biology labs have harder time making them and characterizing them. But with carbon nanodots, it's very easy. So yes, I believe that like everyone should be able to figure it out.
Okay, so to produce these nanomaterials is expensive. So what's the cost? So
yeah, so carbon dots use glycerol and PEI, polyethylenimine, which are quite like cheap materials. You could buy from Sigma, like I think 250 grams of it is $40 or something. So they are very affordable for like a lot of studies that we do. I guess we'll need to do more economical analysis for like field level studies if you were to spray, but we think them as ways to do like transformation in the lab, get your seeds and then put the seeds into the field, like typically how people would do crop improvement rather than directly spray them in the fields, just because that would require a lot more material. It will be probably less efficient. And then we also need to worry about like environmental accumulation, fate of these nanoparticles. So probably those are less desirable applications, but for definitely lab level studies, it is very cheap to make these particles.
Thank you.
If there are no questions, let's thank Wednesday again. And
thank you.
Yes, it's all for today. Yes, we so we can. Today's talk is going to the end. Yes. Thank you to all the finalists today. Thank you to the science committee members for your stimulating questions, and also thank you for all the audience from all over the world. It's quite late here in China, so good night to those in Asia region, and also a good day to those that are in other regions. So we look forward to more wonderful presentations tomorrow. See you.
Thank you.
Bye.
Thank you.
Yeah, good night.
Yeah, nice to see you, Libor.
Okay,
Yeah.
bye-bye.
Thank you. Bye-bye.
Bye-bye. See you tomorrow.
Thank you.
Thank you.
Hope to see
Bye-bye.
you tomorrow again. Yeah.
Bye-bye.
Bye.